extraction and post-extraction. So this is our experimental setup. We had 3 different tanks. 20 urchins were divided between the 3 tanks. In the first tank, we had a control, there was a divider between that and the other 2 tanks because those contained the predator, which was a sea star. And then also in the 3rd tank, we were looking at synergistic effects of both a predator and low salinity. So this is a picture of the actual setup. The experiment took place over 5 days. First we had 1 day of acclimatization so that the animals could adjust to the environment since we're looking at stress. We had an inch of sand in each tank and we scattered shells throughout, and we were looking specifically behaviorally at the number of shell fragments that the urchins used to cover themselves, as well as the movement of the urchins. So we had 2 water changes on day 2 and day 4, and each urchin was fed about 3 algal pellets per day.
We also had clams that were in there in each one as well. Those were for the sea stars, hoping that they would go and actually eat the sea urchins.
So we did have a total of 20. So we had 6 in the control and then 7 in both of the experimental groups. And we had 2 of the purple sea stars, what we used. So now we're going to talk about the video analysis that we did, looking at the behavioral responses of the urchins.
All right, the plan was to record salinity, temperature, the number of shell fragments that were collected each day, and movement over time. Well, what we did was, with salinity, we just made sure to maintain that we had 20 parts per thousand. That was our low salinity tank, and about 30 parts per thousand for the normal tanks. And temperature remained constant. They were in the freezer. Number of shell fragments, we kind of found it was actually very difficult just to count because each one was different size and the amount. In fact, what would have been a lot better to do was actually to calculate the percentage covered of each urchin, which would have been a lot more effective. So, and also, since it was over the weekend, we didn't have a couple of days of shell fragment data, so we just kind of enrolled that for this project. We did look at movement over time, though, and how we did that was we decided to go ahead and assign numbers to describe how each urchin was moving during different hour intervals. So 0, 1, 2, and 3. Basically, if you give it 2, there was 3 to 4 urchins moving within that hour. That's how we did it. It was only over at the 10-hour time frame. I couldn't access the zip file containing the majority of video, but I was able to do this 10-hour footage. And what's interesting about this is that it's the acclimatization period within there. So it's actually, there's no difference in temperature or in salinity at this point in time. So, all right, we can. This is kind of what you're looking at here as they're running around their tanks. It's only 15 seconds. I know. So much more lively in this time lapse of 10 hours, right? So that's 10 hours, they move a whole lot, but no, go away. Next. Okay. So what I did was I went through initially and was like, okay, I'm going to diagram the movement. I'm going to find out where these urchins are, and I'm going to find out at what hour where the majority of the urchins are located. And so what I did is I went through and for the predators didn't move the entire time. They kind of just stayed where they were, and you can barely see them if you look in the video. And basically 1 through 10 it goes to what hour where the majority of sea urchins were basically placed within the tanks. And I also went through after this. I went through and checked out their movement. How are they moving? Where are they moving in the tanks? And I just kind of followed them. And basically this is the generalized pattern of what they were doing. Initially, they were going around the tanks, they would meet the predator, and they would move off. And they would basically start circling back around, and they just kind of stop, and they kind of, in the predator and low salinity, they kind of huddled in the left-hand corner, and same as the control. And in the predator only, they were kind of scattered all about. They didn't really know what to do. Kind of interesting. So the results were, I went through it, I labeled each one on how many urchins through each hour segment that I could see moving. And this is what we ended up having as our graph. And as you can tell here, if you plot this, you can see that the control, after about 2 hours, they actually stopped moving. They just kind of huddled in the left-hand corner and didn't really do anything else for the remainder of the 10 hours. Whereas the predator and predator salinity continued to move. It's interesting to note, though, that the predator actually had more movement than the predator in low salinity. It's kind of interesting, because you would expect the more stressors, maybe it would be higher, but that makes sense. So,
now we're going to talk about tissue extraction. We did use qPCR to quantify the mRNA that was extracted, and we also used the Western.
right, so this is my part of the project. I was looking at the sodium-potassium expression of the cotransporter ion pump. And so basically what this does is basically maintains the sea urchin in an isotonic environment with this environment. And since it's a marine osmoformer, it basically has the same salinity as the environment that it's in. And this is important because if you take the sea urchin and you drop it into a hyper or hypotonic environment, it has to regulate with that environment. Otherwise it will die eventually because it will swell or loss of water. So the primers, I ended up using a post-related species. As you can see is the, you can give it Strongylocentrus purpuratus, I think is what I'd say that. So qPCR is what I ended up doing. Look at this expression, and my efficiency of qPCR reaction, I guess, was very low, 0.76. That's pretty, from what I'm told, that's not good. And my p-values were actually pretty high as well, so that would mean they're not significant, over negative 10 the hour. So explanation of why this possibly be was maybe it's the quality of RNA, since we had a efficiency of PCR reaction was kind of low. Low quality of samples. Use tube feet instead of cutting them all open and taking tissue out of them. There's possible contamination within mine because there was multiple melting points. So not sure, maybe you did something. Samples or who knows. So the results that I found were actually the control here. Just so everybody knows, I multiplied the control by 1,000 so I can even get it onto the graph because otherwise it's basically invisible. And what you see here is that the average expression is higher within the predator-only group than it is with the predator and low salinity. And I thought that was interesting as well because my hypothesis was that the predator low salinity, the increased amount of stressors, you would have increased expression. And in fact, there's higher expression with the predator-only than it was with the predator and low salinity. That's what I was talking about. So just kind of the interesting fact here is talking about what was going on with the sea urchin and low salinity. And we're going to talk about the conclusions possibly why this was, but basically the sea urchin was dealing with multiple things at one time. It was dealing with this predator presence in its tank, as well as trying to maintain isotonic conditions. So it was swelling with water. So what did that indicate? That indicated for me, this suggested that green sea urchins are better able to deal with single stressors than they are as multiple stressors, which makes sense, and that the multiple stressors lower the overall ability to combat stress. That's what I was able to take from that.
So now I'm going to talk about the green sea urchin HSV70 gene expression. We all know what HSV70 is, but I just noted that it belongs to a family of highly conserved proteins that act as chaperones to stabilize and refold the denatured proteins. It is expressed in animals that aren't exposed to stressors as well as ones that are, but it's hypothesized that it will be elevated in animals that are exposed to more stressors. So it's used to protect the organism from endotoxic shock by decreasing mortality, and it's important because it's an indicator of present environmental stressors. So for my experiment, the efficiency was very high. However, the detailed statistics were extremely high as well. So, I mean, the control in low salinity predator tank had a p-value of 0.85, which is very high. This could also be because of the low quality of the tube feed. So here's my graph. My hypothesis was that HSV70 would be the lowest in the control, higher in the predator, and the highest in the synergistic tank due to multiple stressors causing the levels to be elevated. However, what I actually found was that although the control did have the lowest levels, actually the predator tank had the highest. So kind of went against what I thought. Here's just the specific values that was found. And then we also looked at protein, which Ego's going to talk about.
Yeah, that was my part of the research. Who can tell me what is HSV70? We went over this a few times. I just want to get you guys to know this. What is it? Exactly. Why are they important? Is because one of their primary functions is to provide protection to cells and tissues, and protect these cells and tissues from potential injuries as well. my graph, as you notice, the control didn't have any HSP70 protein expression at all. Predator and predator low salinity, these two groups have had the same HSP70 protein expression. So back to the Western blot procedure, which I did. Originally I had 6 samples. One sample, 2 samples per each group. So one sample had gonad tissue, and one sample had digestive tissue. And surprisingly, none of the gonad tissues had any HSP70 protein expression, just the digestive tissues. And they were found in the predator and the predator low salinity groups.
So now we're going to conclude with our conclusions. What we ended up doing to tie everything together was we assigned numbers according to how much expression there was. So this is supposed to tie the genes and the proteins of the HSP70. So we had 0 means that it's not present, 2 means it's present, and 3 means it's present in high quantities. So we can see that the control had the lowest expression in the gene expression, but no proteins were actually translated. For the predator tank, there was the highest amount for the gene expression. However, it did drop a little bit in the proteins, and then it was the same for all of the predator and salinity. So our first conclusion was that the addition of stress increased both the sodium, potassium, and the HSP70 gene expression. This isn't dependent on the type of stress; it happened for both kinds. And our explanation is that both genes could possibly be activated by the general stress response.
Yeah, I just thought it was very interesting that it was kind of a generalized stress response. As you can see, if the sodium gene expression was also increased, it was the highest in the predator only when it was maintaining the same isotonic conditions as the control. So maybe that would suggest a general stress response there.
We also found that there was the highest rate of gene expression in the predator only as opposed to the predator and low salinity tank. And our explanation for this could be that there was kind of a lack of multiple stressors, and so they were able to devote more energy to the transcription. So if there's multiple stressors, then kind of the animal would get overwhelmed, and they wouldn't be able to put as much energy into each specific response.
And here's something that's interesting, and we're just kind of going on a limb here. And from Igor's presentation, what he found out was the bands were pretty much the same.
Light, but they were much lighter.
Yeah, exactly. So we're just kind of making the assumption here that they're going to be somewhat, if we were to take them out and actually quantify maybe somewhat of the same expression. So that's suggesting that the HSP70 protein production is maximized in both tanks. And what we're saying that is because as the expression increased, it still maintained the same amount of detectable protein levels. And so that's there. And basically this suggests that urchin exposed to multiple stressors have a decreased survivability rate. And why I'm suggesting that is because the increase in stressors increases the overall mechanisms involved, which decreases to the overall gene expression. And that can be seen there in the predator and the salinity. So I kind of put this out there as an example of what happened with a single stressor. The urchin is expressing the sodium, but is in the isotonic conditions, and it may not be translating the mRNA. Whereas in the multiple stressors, it actually is expressing it and must translate the mRNA in order to basically go back to isotonic conditions. And there's also like unknown effects that we don't really know, which kind of led to, this is kind of an overview. And it must produce the proteins and sodium compounds to maintain isotonic conditions, but it also must regulate other mechanisms